Hello, this is Rupinder Syal and welcome to Spartan Tutorials. Today we are going to talk about three different technologies which are based on the same principle, and that is the chromatin immunoprecipitation. These related technologies are highly useful and are currently used on an extensive basis to characterize protein DNA interactions, whether it is the binding of transcription factors or DNA replication apparatus or other DNA binding proteins to the DNA. These technologies are really helpful in characterizing genome-wide analyses of binding of these proteins to the DNA. So let's take a look at these chip-based technologies. Now, chromatin and nucleus is a very crowded place, as you can see in this schematic, because we don't have the actual structure of the whole DNA inside the chromosome, but we have some idea that it is a highly structured place. In the normal interphase, when the cells are doing their thing, that is they are not dividing, they are present in this loose chromatin form where they are associated with protein and DNA. So we have DNA and protein complex, and this is basically referred to as the chromatin. And these include basic nucleosome level organization of DNA, as well as higher structures of DNA which lead to more and more coiling, about which we are still getting some knowledge. There are several models of hierarchical DNA organization, like the solenoid model or the zigzag model. We are still pretty far away from the the complete picture of how this nucleosome level organization leads to the formation of the chromosome. But anyhow, we know that this chromatin is present. Okay, this chromatin is there. The DNA is bound up in all these proteins which are highly positively charged, helping in wrapping up the DNA. And the proteins which need to express some genes, for example, some genes are expressed as a result of a viral infection, or some genes are expressed as a result of embryonic development or differentiation. These proteins usually are transcription factors, and they bind to the DNA sequences. Okay, for example, shown here in shades of blue and green, they bind to the DNA, they open up the chromatin using chromatin remodelers and other enzymes which use ATP to basically tear apart the DNA from the proteins, and then they transcribe as well as turn genes on and off. So this is a fascinating area of gene regulation, and previously there was no handle as to how to approach this. There had been a lot of studies like the lac operon and other operons, showing that DNA binding proteins exist, like the lac repressor and the lambda repressor. And even for yeast and other eukaryotes, for example, GAL4 DNA binding proteins were identified in the early 70s and 80s, but there was no systematic way to actually probe how and where the DNA binding proteins are actually binding. There were some in vitro techniques that we have covered in our videos past, for example, DNA footprinting, gel shift assay, but on a genome-wide basis and in a more of like an in vivo context, we did not have such techniques, and that deficiency was fulfilled by the development of chromatin immunoprecipitation. And that is the technique which is referred to as the ChIP, chromatin immunoprecipitation. So it is referred to as ChIP. So kindly keep in mind this capital C, small h, and I and P are all capital, just H is small. Okay. Here is an example of lac repressor binding to the DNA, some canonical examples of DNA binding proteins. We have beta interferon enhantiosome. These have multiple binding proteins, NF-kappa B, IRF7, IRF3, other proteins, June. They bind to the same interferon enhantiosome and regulate gene expression. And then we have really big assemblies, like here we have the gigantic mediator and RNA pol 2 pre-initiation complex. Each different color is a different protein, so you can imagine this is what is happening inside the cell when a protein is being expressed, when a gene is being transcribed, and this tiny thing, this is our DNA, and the whole truck on the top of it is the protein complex, So we need some really nice ways to actually tear apart the interactions and the sequential loading of these proteins to the DNA. So the origins of ChIP or the chromatin immunoprecipitation go way far back. This is, this was pioneered by John Liss's group at Cornell, where they developed cross-linking techniques where they cross-linked using ultraviolet rays protein to DNA and then analyzed the positions of these proteins on the DNA. Okay, so that was the earlier basis of chromatin immunoprecipitation. There was no amplification step or way to know where in DNA they are binding. The only thing that they could do at that time was southern blot analysis. So remember, this is 1980s. PCR is not in popular use, and widespread use of PCR started all after 1990. Okay. So this is the overall idea of chromatin immunoprecipitation. We have DNA protein cross-linking. So as I said earlier, in the 1980s by John Liss's group, this was done with UV. Later on, we used formaldehyde. So researchers adopted the use of formaldehyde. The advantage of formaldehyde is that the cross-links formed between DNA and protein are reversible. You can heat the cross-links and they will be reversed. They will be terminated. So we cross-link or bind protein to the DNA. We basically press a pause button on the DNA protein interaction. Then we lyse the cells, okay, break open the cells. We sonicate, so this is using the sound waves to break and shear the DNA so that the larger DNA chunks are broken into smaller parts. Or we do restriction diation, that will also serve the same purpose. It will break down the DNA into smaller chunks. And we are left with fragmented chromatin. Okay, this fragmented chromatin, which has some DNA binding proteins bound to them. And then, for example, we are interested in where our protein X binds. So what we will do is we will have anti-X antibodies, and we will use that to precipitate this DNA protein crosslink out of all the DNA protein crosslink that has been obtained. So the specific genomic fragments where the protein X binds will be picked up by the antibody along with the DNA. Now you will reverse the crosslink, you will digest it with the protease, so to remove the protein, and you are just left with DNA. And that DNA can be amplified by using PCR if you are already, have some idea about where the protein binds. You can also analyze it by qPCR, quantitative PCR. You can hybridize it to microarrays, to a tiling array, for example. This is called ChIP-Chip, and that we will discuss. Or you can just sequence the whole thing, and that is called ChIP-Seq. So this whole thing is common. We can do PCR or qPCR, and this is referred to as ChIP or ChIP-PCR. Then we have microarray, and this is called ChIP-Chip, or sequencing, which is called the ChIP-Seq. So these are the three different variants. So they were actually developed along a timeline. First ChIP and ChIP-PCR were developed, then ChIP-Chip came along when the microarrays were developed around 2000, 2005, 7, around those time, and ChIP-Seq was developed around 2008 and 9, and subsequently even more refined versions of ChIP-Seq are available right now. Alright, moving along. So here is one example of where we can look at the actual data produced by a typical ChIP experiment. Here they are looking for the binding of Tata binding protein, TBP, one of the proteins involved in transcription. It binds to the Tata box, and they are looking at its binding at three different regions. One is a GAL4 open reading frame region, and these are the location of the primers that they will use for PCR. So definitely Tata binding protein is not going to bind here, so they should get no signal, right? So this is their negative control. Then they have ADH1 or alcohol dehydrogenase 1 promoter, and this is their Tata box, and this is the location of their primers. So of course it should amplify, and I should say ADH1 is a constitutively active promoter, so it should give a signal nonetheless. And lastly, they have an induced promoter, which is the GAL1, and the primers are similarly located around the Tata box, so it should give signal only when you add galactose to the medium, according to what we know about the galactose operon. Okay, so here we are looking at without formaldehyde treatment. What are they looking at? So they are looking at PCR of these 3 fragments in the input when galactose is used, but without formaldehyde. So remember, no DNA protein cross-link, so there is no TBP here. Okay, so it should give the PCR product. Okay, right? They are not doing any IP here, this is just their input material. But when they do IP with anti-TBP, because there is no formaldehyde to cross-link it to the DNA, there is no signal. So no, no, and no. No signal at all. Now they go for the different inputs. So here they are different inputs, low input, high input, just to show how much amount of stuff they have got. In presence of glucose and galactose, so you can see huge amounts of DNA being amplified from these regions. Okay, that's normal, that's just their input. They have not done their IP yet. Now we will see their IP experiment. Okay, here we are looking at their IP experiment from pre-immune. Okay, so this is before IP. So of course no signal, just like here, in presence of glucose or galactose. And when they look at IP with anti-TBP antibodies in presence of glucose and galactose. Now see here, GAL4 ORF, pretty weak signal, and it does not give almost any signal, as compared to what they had here. Okay. ADH1, which is constitutively active, it gives a signal in both, right? And GAL1, which is stimulated only in response to galactose, it gives response only in the galactose lane and not in the glucose lane. So this is a very good example of how ChIP can eliminate whether your protein of interest binds to a particular region or not. Now we come to the ChIP-Chip. So ChIP-Chip was, the next logical step, where they used a tiling array or some sort of array where they had huge amounts of DNA sequence from the whole genome when the whole genome sequences were available. So this is one example of Fe matrix gene chip array. There are multiple types of these arrays, but the basic idea is the same, that you have a whole matrix, a whole columns and rows of different DNA fragments loaded on this small chip, which is very small size chip. You can see comparison to the matchbox match here, that it is a pretty small thing. And what you are looking at is hybridization of labeled mRNA to this chip, okay, or labeled DNA. So let me just erase all this. Maybe this will be better. So this is how ChIP-Chip works. Same protocol, add formaldehyde, sonicate the DNA, make it into smaller chunks of about 1,000 base pair, add your antibody for immunoprecipitation. Okay, immunoprecipitation will happen. Okay, and reverse the crosslinks, purify the DNA, and add the label. So one of them is green and one of them is red, and the combination, if they both hybridize to the same, gives a yellow signal. Okay, and here they are not looking at any IP. Okay, so they are just labeling it. So this is their control experiment and this is their actual condition, and they are looking at hybridization and further amplification of the signal. So this gives them a very good genome-wide, although not completely genome-wide, but a sufficiently genome-wide, maybe just all the ORFs or a specific chromosome level analysis of where a particular protein binds. So this was a significant advancement over the previous ChIP studies, where they were looking at one locus at a time. And finally we are, the whole shebang, ChIP-Seq, where we just sequence the hell out of it, right? So similar principle, we have sample fragmentation and immunoprecipitation. Okay, pretty same thing. And here we are looking at different types of IPs. So people are also looking for example, individual proteins like transcription factors, and even looking for histone modifications now. It was also done for ChIP-Chip also, but in ChIP-Seq this was really coming forward, and people were appreciating the role of histone modifications on a genome-wide level. Okay, so this was a big advance. They purify the DNA in the next step, and now there is a ligation mediated PCR step. A ligation mediated PCR step is necessary because the amount of DNA that is recovered for immunoprecipitation is really tiny. Although you need only a few nanograms and few micrograms to do this experiment, but still you have only a few nanograms to start with. So what happened is, in this protocol, a ligation mediated PCR step was added to amplify the amount of immunoprecipitated DNA. The same ligation mediated PCR step was also used for ChIP-Chip analysis also. And then you do different types of next-gen sequencing, either Illumina or Roche sequencing, or ABI, Applied Biosciences, or Helicose, any type of next-gen sequencing methods to get millions of reads, and then we map it onto the genome. Here you can see one example. This is from the earlier studies. You see here we have the NeuroD1 gene and the NRSE. This is the transcription factor. So they are looking at binding of this transcription factor on the NeuroD1 gene involved in neurogenesis. Okay, and you can see that these in red are the number of reads mapping to the genome. So it gives a nice peak basically. So you will get, for example, a snapshot of the whole genome where you can see the peaks of the DNA binding of that particular transcription factor, which will kind of indicate where the stronger binding occurs. Okay, and there are different algorithms, different peak calling algorithms which identify the peaks. For example, is this a peak or not, or is this a peak or not, comparing it with different controls and multiple replicates. So there is a lot of statistical as well as computational analysis involved in here, but the take home message here is you can identify binding sites for different factors, or you can identify histone modifications on a genome wide level using ChIP-Seq. Here is another example from a data from a paper where they were looking at genome-wide maps of chromatin state in the pluripotent cells and the lineage committed cells. So here they are looking at two histone modifications, H3K4 trimethylation and H3K27 trimethylation. H3K4 means histone 3, lysine 4, and 3 methyl groups are added to the 4th lysine. And this is deposited by the trithorax group of proteins involved in gene activation. And H3K27 trimethylation basically means histone 3, lysine 27, and trimethyl group is added. This is deposited by the polycomb group of proteins, and this is involved in gene silencing. That's why it's labeled in red here and green for the trithorax H3K4 trimethylation. So here you can see the snapshot of the chromosome. This is the mouse genome, okay, around the 300 kb region. So you can see many different genes running either on the 5' to 3' or 3' to 5', and you can see for some genes, for example, here this gene is active, whereas it does not have any inhibitory mark, and some genes, they have both the marks. For example, this gene right here, it has the inhibitory mark as well as the activator mark. So these were labeled as having the bivalent marks. So probably they are what are known as poised for expression. They have the inhibitory as well as activation marks. Okay, so this gives you a good snapshot. Nowadays, you can find multiple maps of these histone modifications as well as transcription factor binding events for almost all the well characterized model organisms. You can do analyses, comparative analyses of these genome-wide binding maps, and they are readily available from UCSC Genome Browser as tracks. So you can do very good analysis as well as hypothesis testing using these genome-wide binding maps. Here you can see another study. So this gives you an idea about the dynamic nature of ChIP-Seq. This is looking at the Zelda binding. Zelda is a transcription factor involved in the maternal to zygotic transition in the early fly embryo. So when the maternal genes, they are basically switched off all the time, when the embryo starts to develop, and the zygotic genes are turned on. In that transition, Zelda plays a critically important role. You can see the early division cycles of Drosophila here, and Zelda basically is not here at this HMGZ gene, but around nucleocycle 13 and 14 it appears. And you can see at CG18269, earlier it is here, but it is diminishing in its occupancy around cycle 14. So this gives you an idea about the dynamic nature of binding of these proteins. Okay, so very good technology, and further refinements are continuously being made. However, there are a couple of limitations of ChIP-Seq as well as other chip analyses, and there are 3 of them I would like to bring your attention. One is that usually ChIP-Seq and ChIP-Chip, they have really low signal to background ratio. Compared to the negative controls, they give very low signals, so that can be an issue. It is difficult to determine the precise binding site of a protein because their resolution is about 300 base pair, so you don't exactly know where your particular DNA is binding. You just know the kind of window that, okay, in this 300 base pair somewhere is my protein. Although I would like to mention that there are finer and better techniques available, which really narrow down the binding site, exact binding site occupied by a protein. And finally, just because a DNA binding protein is present on a DNA sequence does not mean it is functional or it is doing anything. And that has been a big issue. There have been a number of reports showing that these transcription factors, they bind promiscuously in the whole genome at even inactive regions or where they have no function whatsoever, but they do bind. So determining the causality or the functionality of binding of these proteins to the DNA is kind of tricky and has to be supplemented by other functional assays. Okay, so those are some of the limitations of ChIP techniques, but overall I think this has been a revolutionary technology in analyzing protein DNA interactions on a genome-wide scale. Okay, so that was my discussion of ChIP, ChIP-Chip and ChIP-Seq technologies. If you have any doubts, comments or questions about it, please let me know. Thank you for watching, and I will see you next time.